a trajectory. In a decade, we will have these data sets that you described, right, for the United States version of the biobank, and even more from biobank, of course, in that time frame. And yet, even along the points in this trajectory, what are we confident to be able to do now, right? As you said, we can deconvolute, complex phenotypes to find new profiles for attacking a disease phenotype reflection that we think will be productive in human patients. Yeah, that's very nice. And of course, we will have more confidence, in those predictions and probably greater efficiency in addressing them when we have more of this data. But in the meantime, it seems like what do we think we will be getting kind of in the near term, that we will have truly new targets, right, that haven't been addressed before, or a better understanding of how to modulate existing targets, or mutated forms of those targets in a subset of the population. And at what point will this mean that we will really be able to execute on personalized medicine, because we'll have enough patient data to actually design these discovery pathways for specific patient subsets in a forward sense. So what are some of the things that we think we can do now? What are some things, 3 years from now, and what are some things that after this decade?
It always wonder, but today, if you look at the actual pictures, we have thousands of identified targets as being implicated in a disease, yet they are not addressed by pharma at all. They are so peculiar. Let's say the generic word is undruggable. Undruggable because you cannot reach them, or undruggable because there's no way you can find any ways of finding molecules that can inhibit them or modify their specific working cycle in a disease. So first of all, as a part of the society, at what point are we going to look for other targets? Well, we have those, and we maybe try to find specific tools and means of solving this issue. We have them. They are here, thousands. We discover many more using these new tools. Are they going to be put in the same pool of undruggable? Is it just another set of data with their own problem? Where is the balance in between, right? Sometimes the problem is not identifying a drug, but knowing how to take it all the way through a cycle to find a target, but to pull it all the way to find a specific molecule that will inhibit it or interact it. So the balance between the two is that where we can put your money in, right? Because it all comes down to that, right? And the shift. So yes, identifying that target, maybe proposing them, and then what?
With the existing targets, as you describe them, we will better understand them, through these processes. But also, there are techniques now that we are able to better annotate these existing targets in a dynamic sense. We're able to think about them in different ways, both computationally as well as experimentally, in ways that opens up more ideas about how we may address them, from novel pockets to tagging them for degradation, or tagging them for complexation in a different subcellular or position, which leads to different activities. And these are also coming about, I think, through our understanding that increases through our just basically general learning of these processes.
Yeah, there is a trend. So I was mentioning the undruggable. There's a trend that now for a few actors that are trying to act on this issue of undruggable. Most of the time, the cycle is started when you have a target and you have an idea that comes either, or even data that comes from experiments, crystallography, you're naming it, NMR and so on, and you have its structure, so you can start playing by trying to find a drug. But we all know that targets and molecules in general are dynamic, right? And we have only one view, we have one picture, and we try to influence that picture that is probably not the one that should be addressed in a working cycle of this molecule. So strategies today are to try to enable using techniques such as AI to go and fetch for these hidden conformations that are hidden, also cryptic pockets or allosteries, for which now it could become possible using exactly the same techniques as today to find a binder, PROTACs or small molecule or anything that you wish. So there is a lack of knowledge about targets. We know so little about them. One fold, maybe 2 or 3 folds, minimum energy conformations. And what we need is to uncover all these dynamical transitions that they have and target this hidden conformations that are today, playing a role probably in the working cycle of these targets. The trend now is to produce data that will generate these conformations. Now, said in Nebula, we even have build up a complete new strategy based on AI to uncover these conformational changes, and it opens really the world for exactly same target that is undruggable, no cryptic pockets. You don't even address it as a pharma, even if you have the best machinery for drug discovery. It's just because it looks like a softball, it's like a tennis ball. But when you apply the technology that we have developed, you uncover these cryptic pockets, and now you can start identifying drugs and ranking them and so on and so forth. So this is huge because that means about the few thousand, 80% basically of known targets applied in a disease are undruggable, not addressed. 80%. It's huge. So first, it's a free patent space. Nobody knows what they are, and if you can start playing with them, it's much better. So these are the things that we put as target identification, but not just the sequence on one form, but the whole dynamical space, in conformational space in which it evolves in a realistic condition in a working cycle.
In this case, will you be able to test your compounds if it's really linked to this complexity? If you purify the proteins, will you reach the same level of modification, interaction, complexity of the change of the shape of your target? Because what I imagine it's basically we do the target validation in silico, you design, you use all the tools for ADME and everything, and you optimize your molecule only in silico, and then at the end you just jump directly into a cellular models, classical or advanced cellular models, and then the next step is the animal model. And so is it something, it's where you project with these undruggable targets if we can't have any biochemical tools basically to study them?
No, it's the same, except that if you are trying to find your drug, start with a minimal low-level docking, right? The fact that you have several conformations, then your target is not one single structure and you start your pipeline. It's 10 different pipelines with the same molecule that have 10 different conformations. And maybe conformation 1 will give you the best shot, but conformation 7, in which you have a hidden pocket, is the one that gives you the best compound that is linked to it. And then you test that experimentally, because in the bench, what you guys do is that you are dealing with a molecule that is undergoing these changes. It's undergoing this conformational change. So the only thing that is very important is that we put physics-based modeling in the original AI-assisted engine to be able to produce realistic conformations.
Yeah, and I think another thing that this conversation has kind of pointed out is that drug discovery has been often a reductionist approach, right? You're always starting with a target, which sure is a single protein which has multiple conformations, but most diseases are probably multifactorial. And our job to date has really been to focus on that single target, find a drug that's safe and effective, but usually they're not perfect in solving the problem of the disease. So I think this opens up more ways to think about how to solve the problem.